Thank you, Albino. So after these last two talks that focused on patients and on brain systems, I'm going to ask you all to narrow your focus a little bit until we reach single neurons, and in fact, even more so, the individual synapses on the neurons. You can see here these postsynaptic spines scattered along the dendritic tree. Each of these tiny protrusions is a site of contact between this neuron and its thousands of upstream partners. It's at these synapses where, at the site of these synapses, we can have changes. This is called synaptic plasticity, and it's a very, very well-studied phenomenon for the reason that its synaptic plasticity changes in strength that can change the network, which we think are the major sources of memory encoding and memory storage in the brain. So in a meeting such as this one, where we're focusing on neurodegenerative disorders, and one of the most devastating symptoms can be compromising of memory, it's very interesting for us to look at the healthy system and see how it functions, to give us more of an idea of what could go wrong in disorders. If we focus a little bit more on one of these dendrites, I'm going to lead you through some different types of plasticity that can happen here. The most widely studied form of synaptic plasticity is long-term potentiation, or LTP. And this is going to constitute a strengthening of this synapse here. LTP has some important characteristics. For a start, it's input-specific. That means due to activity which happens in a correlated manner between an upstream partner and this downstream part of the synapse, it will strengthen this particular synapse, leaving the other ones around it unchanged. What this means is that you can have individual changes to synapses along the whole dendritic tree, and in fact along in a whole circuit, neural circuit. And it's these thousands of changes that actually lead to the information being stored. That's why I've called it here information encoding plasticity. But this is not the only type of plasticity that occurs in dendrites. There's another type, which is in fact the main one that I'll focus on today, and we call this homeostatic plasticity. And this has some different characteristics. So instead of being an input-specific mechanism, it's actually a more global mechanism. It happens along dendrites or along in the whole circuit. And as the name suggests, the homeostatic part, instead of being information encoding, we think of it more like a stabilizing type of plasticity. So if, for instance, excessive LTP or its counterpart, LTD, long-term depression, pushes a neural system outside of some intermediate optimal bounds to leave it to become, for instance, with many strengthened synapses and an overactive circuit, this mechanism, homeostatic plasticity, can bring the circuit back down into the optimal bounds. So the questions that I focused on during my PhD were related to homeostatic plasticity, about which much less is known than about LTP. The first one was, what are the structural correlates of this type of plasticity? So does it actually change these structures here on the neuron? And the second one was, how can these two types of plasticity, LTP and homeostatic plasticity, interact together in a neural branch? I'm going to tell you about some of these results today, and if you're interested in some more, you can come to my poster. It's number 70 later on. So this is the technique that I used. I used organotypic hippocampal slices, and these. So to introduce homeostatic plasticity into these, I did a chronic manipulation of activity levels. In this case, what I used was a chemical called TTX. This is a sodium channel blocker, so it's going to stop all neuronal firing in the slices. So these slices, which were incubated for up to 48 hours in TTX, were compared to control slices, which underwent normal levels of endogenous activity. So first, they verified that indeed this activity blockade caused homeostatic plasticity. And what we would expect is after long-term chronic activity blockade, synapses would be strengthened. And indeed, by recording these mini APSCs through electrophysiology, what we could see was after 48 hours, these TTX neurons, the synapses were significantly stronger, shown by an increase in amplitude. And in fact, when I plotted the control MEPSCs against the TTX MEPSCs, you see that there's been a linear scaling upwards of the synaptic strength, showing that we've induced homeostatic plasticity. Next, we wanted to see, does this have any structural consequences for the shape of the spines, for the size and the shape of the spines? So using two-photon imaging of dendritic branches and measuring the spine volumes, I looked to see whether there are any changes throughout this period of activity blockade. And what you see is after 48 hours, indeed, there is a significant increase in the spine volume after homeostatic plasticity. Now, when I plot the control spine volumes against the TTX spine volumes, you see there's also been a scaling upwards, although in fact this now is not a linear scaling, but superlinear. This has some interesting implications about what the differences are between the functional side and the structural side of the synapses, which I don't have time to go into now, but do come talk to me later if you'd like to talk more about it. So now, having defined what is the basis of structural changes in homeostatic plasticity, we decided to look at how does this interact with LTP. To introduce LTP, which, as I told you before, is a local mechanism as opposed to global homeostasis, we use this technique called glutamate uncaging, whereby using a laser light, I can activate individual synapses. So I can induce LTP only at a single spine, leaving the rest unchanged, as you see here. So first of all, I wanted to know, can spines that have undergone homeostasis even undergo more LTP? Is it even possible to have more plasticity? So the graphs I'll show you will compare this individual stimulated spine here to the non-stimulated neighbors. And what you can see is comparing the TTX and the control here, indeed we do see LTP. So even after homeostasis, the spine can undergo more plasticity and can, let's say, encode more information. If we look closely at these two curves, what you see is, so this is the immediate post-stimulation response, and then they settle down to a steady state level. If we look closely at the end of this, however, what you see in the control case, by the 2 hours after the stimulation, there's actually no difference between the non-stimulated neighbors and the spine. But in the TTX case, there's still a significant difference. So it seems as though, in fact, what we've done is sensitize the circuit to allow the homeostasis has allowed it to express more longer-lasting LTP. To look at this further, we decided to characterize the spines by their size. Spines along a dendritic branch can exist in many different sizes. You get some very large and some very small. So I split up the spines that I stimulated into large and small and plotted their time courses separately. What you saw, first in the large spines, there was no difference between the TTX and control. But in the small spines, you see something different. So actually what you can see is the steady state level of LTP here is higher after TTX application. And this means that there's a specific enhancement of LTP after homeostatic plasticity. And so the last result that I'll tell you is actually not even to do with the stimulated spines itself. It's to do with the neighboring spines here along the branch. I told you that in normal circumstances, the LTP induced by this technique is input specific, so it changes only the stimulated spine and not the others. But is this still true after homeostatic plasticity? So to understand this, I split these neighboring spines into two groups. Those nearby the stimulated spine, or up to 5 microns, and those far away. And what I found was after homeostatic plasticity, unlike the normal case where there's no change, we actually see a strong effect of the pre-application of TTX. See, these nearby spines actually show an increase in size after the stimulation, and they level out to a higher level than these far away spines. So this implied to us that there's some sort of metaplasticity along the branch, that some factors nearby the stimulated spine are allowing these other neighboring spines to express plasticity too. And this is another, yet more evidence that there's some increased sensitization of the circuit to LTP or to activity after they've undergone long-term activity blockade. So just to conclude, I told you that homeostatic plasticity causes reversible structural changes to spines, that after homeostatic plasticity, LTP at small spines is specifically enhanced, and that metaplasticity after homeostatic plasticity causes short-lived structural growth at neighboring spines. We also looked at many other factors to do with the way homeostasis changes LTP, and there on my poster, it's number 70, so do come ask me about them later. And I'd just like to thank my group, the Neuronal Structure and Function Lab, and my supervisor, Imbal Israeli. Thank you.